Okay, so I'll go ahead and start. This is a session on rapid microbial detection. So what I have here is I'll talk a little bit about what the current method is. Many of us know what the current method is. It's mainly plate counting, or CFU, which was invented in 1887. We still use the same method, where basically we take the water and we put it in a petri dish, and we wait for the microorganism to grow. Once they grow, they form a colony, and that colony is called a CFU, a colony forming unit. We count the number of colonies that are formed in the plate, and that's how we determine the microbial content of the water. Obviously, when you're waiting for the colonies to form, you want the microorganism to have the right kind of condition. They need the right humidity, the right media, the right temperature, the right everything to grow. And that's basically one of the main drawbacks. You have to wait 5, 7, sometimes longer to get your results. What we have today is our online bioburden analyzer. It has a very different way of looking at microorganism. It has a 405 nanometer laser. What it does is it pulls the water inside in a flow cell. And then the 405 nanometer laser basically illuminates the water flowing inside the flow cell. If there is an object in the path of the light, it will have scattering. One, it will have me-scattering from the object. If there is a microorganism present, which always have the metabolites riboflavin and NADH, then it will have the fluorescence scattering. Sophisticated algorithm inside the analyzer basically picks that object which has both me-scattering and also fluorescence scattering and displays it as one AFU. What it basically means is one microorganism floating through the water. If you want to know more, we have our analyzer in our booth 603. Moving forward, today I bring you here a case study, a project we did with Stelmas. We basically had a loop of water, and we had 3 objectives, some of the major questions we received from our customers. The first one, how do I know that there is no excursion? How do I know my regular water should look like? What is the AFU value that I'm looking? I know what my CFU is. I don't know what my AFU is. And that's exactly what we wanted to understand. How does the routine operation in your water system affects the AFU number? The second objective we had was to understand the efficiency and the effectiveness of regular processes like hot water sanitization to maintain the microbial content of your water system. And finally, the third objective we have was to see what is the response of the AFU and the analyzer when there is really an excursion. So we ran this study with the help of Stelmas in their facility. So first one here, what you see in the graph, let me see if I can point. Yeah, I can, right? Yeah. What we have here is a graphical representation of the data. The green line you see here are your AFUs. What you see here are the regular responses of your AFU for routine operations. And I'm going to talk about a few ones. So these are the spikes. The biggest one you see are mainly sanitization. When the temperature goes up, you see those. The small ones you see here are also results from routine maintenance in the water system. How do we know that they were sanitization? Only because we also look at the temperature. The first one I want to talk about is routine operation. The beauty of the analyzer is you have all of this data time-stamped by the second. The data comes in every 2.2 seconds. So you know exactly when you have the spike. And this cyclic pattern that you see here in the data, we were able to understand they were related to hydraulic effects. What were they? When we looked into the processes, what we found was they were mainly tank filling. So when more water comes into the tank, you obviously have more microbial content. This is what we call the signature of the water system. Once you have the analyzer online, you will know the signature of your water system. And that's exactly what you're looking for to understand the regular operation and how your AFU values will differ. The second we had is the sanitization. Here we also see the temperature. The red graph here shows you the temperature. And as you can see, every time there was a spike in the temperature, the AFU values rise. We know from experience with different customers that the AFU spikes up during hot water sanitization, maybe because there's some biofilms which are fluffing away. It's mainly a routine response of the microorganism. When you heat it up, you see that spike. Again, this is something which you will be more and more acquainted with your water system when you have an analyzer online. And this basically told you, if you see in between the sanitization, is my sanitization effective? Should I do some optimization? Can I do my sanitization a week later? Those are the decisions customers are usually able to make from this information. The other thing you can find out, and this is a tank where the water was recirculating, we can see that after each sanitization, your baseline AFU continuously drops, because obviously this is the same water and you're sanitizing it all the time, so naturally your baseline AFU drops. You have more cleaner and cleaner water. This is something you won't be able to see in your CFU analysis when you have zeros or ones or twos and you have the same numbers all the time. And that's exactly what this analyzer brings you. This is the third objective we had. Can I see an excursion when it happens? So what we did was, again, as I said, you will know the signature of your water system. So for this specific water system, the baseline AFUs, as you can see, is around 2K, 2,600 AFUs. That's what the baseline was. During heat sanitization, we can see that the baseline moves up to about 22K or 22,000 AFUs, which is fine. That's what the microbial content of the water is. During other times, we also see that the AFU fluctuates between 12,500 AFU. And then we decided, in a controlled manner, to add some tap water, which is obviously not purified. The second we add the tap water, immediately you can see your AFU go up to 51K. That's an immediate response. You take an action in your water system, you immediately see the effect in the water. You don't have to wait for 5 to 7 days. You see that effect immediately. You know immediately what has happened in your water system. You have added something, that's why you know it, but you can see it that if an excursion happens in your water, you will see it in your AFU count. You will know it from your AFU count what happened. And that is basically the major benefit from this analyzer. It does not depend on the growth of the microorganism. You don't have to wait 5 to 7 days to know the quality of your water. During excursion, you will immediately know what it is. For this occasion, we also did plate counting, and obviously the results were TNTC when other tap water was added to the mixture. The key takeaways from this: real-time online bioburden monitor obviously gives you real-time results. The analyzer is mainly meant for process control. It gives you a better understanding of your process water. It tells you the effectiveness of any changes in the activity during the processes. It shows you immediately if there is an excursion in your water system. And that's exactly what this analyzer is for. You don't have to wait 5 to 7 days. And that brings me to the end of my presentation. If you want to know more about our analyzer, you can scan the code there, and we can have an appointment with you. Now I'll open it up if you have any questions. Can I take any questions here?
Thank you very much. That was very interesting. I deal also with injectables. So can we do the analysis of purified water also?
Yes.
If there is the ambient, if it's running on the ambient temperature.
It is meant for purified quality water and also WFI, ambient. It is also can be used in hot Wi-Fi, but your AFUs will be different. You can use it for purified.
We have to validate it for that.
What do you mean by validate?
No, we have to make the limits for that. Huh? If we are working on the purified water at ambient temperature, or we use the hot water, definitely there would be some change.
Absolutely. When you have the analyzer in whichever loop you have, you can continue to do your CFU counting for a period of time, and you can validate the AFU. As I said, the signature AFU, you will know if you're using it for your purified water, what is the signature AFU that you're looking for.
Okay.
While you're still doing your CFU counting, so you know that your water is under control.
My second question is that what environment this equipment requires? Because in the water treatment plant, there is a hot environment over there. So which environment is required? They need a separate cabinet with having a air conditioning system and low temperature, or we can put it with the water treatment plant?
You can put it in the water treatment plant in Asia, in China, Japan, Indonesia. We have the analyzer in a water treatment plant with different pharma companies and micro-E customers. It can withstand up to a certain level of temperatures, no problems. Normal temperature, it's not air conditioned. We can get that details from our data sheet where it tells you the various things like what is the water loop pressure you need. We need a minimum of 20 psig pressure in your loop, and things like that you can easily get from the data sheet. It does not need air conditioning. It can stay in a normal water room.
Thank you.
Thank you for the interesting talk. I have a question. You were telling that in 100 milliliter WFI, you see 2,600 AFUs, which is quite a large number for WFI. Assuming that WFI needs to be virtually sterile, how do you correlate these WFI counts to real counts? Knowing that I don't expect that this is all viable but not capturable microorganism, because that's really what we would not like to see in this environment. So, are you expecting to have some autofluorescence molecules, particles? What is the interpretation of this count?
Yep. Thank you for the question. I have got this question every time I have presented, yes. It depends. WFI quality water, we cannot really tell you what your baseline AFU will be. It could be high. We have seen WFI, which is as high as 1,000 or 2,000 AFU. Again, remember, when you see 1,000 or 2,000 AFU, that doesn't always convert to your CFUs because not all of them grow, right? For this specific experiment, this was a water loop at Stillmer's facility, so we were using the same water in the loop. So it wasn't really in a pharma circumstances, so this was definitely, I would say, an experimental loop that we were doing this analysis, and for this loop, the signature was about 2,600. And again, I want to bring this out that you do not know what will be your microbial count. It could be 1, it could be 100, it could be 2,000. It really doesn't matter. What you need to focus on is not the number itself, it's more about the baseline, the trend that you're looking at, because that's exactly what you're going to look at. You're not releasing your water based on AFU counts. You're still doing your point of use CFU counting, and as long as your CFU counting is within your limits, you really don't have to worry about your AFU counting, whatever those numbers are. What you're looking at is the trend. If the trend stays in 2,000 while your CFU counts is under your limit, be it that.
Any further questions from the audience? If not, let's thank the speaker for the wonderful talk.
Thank you.